Hi, my name is Ria DeVaker, and I'm a senior neuroscience major at Drew University. Today, I'll be going over the paper, Antidepressant Actions of Ketamine Engage Cell-Specific Translation via EIF4E by Aguilar-Valles et al. So, antidepressants are medications that are used to treat depressive symptoms. The most commonly prescribed antidepressants are known as SSRIs. However, they don't work perfectly on everyone, and they take a long time before an effect is seen. Previous studies have found that ketamine has antidepressant properties. This trend was first seen when ketamine was given to suicidal emergency patients as a sedative. They found better outcomes in patients administered ketamine. Ketamine is normally used as an anesthetic, but this finding triggered a series of more studies. At subanesthetic doses, ketamine can rapidly help alleviate symptoms of depression. However, it takes a long time before a new drug can be approved, and simply showing one result isn't enough. There needs to be more data on how and why this is occurring before a treatment can become widespread. This need is compounded by the fact that ketamine has a reputation of being used as a recreational drug as well. So overall, this paper established how ketamine is able to work as an antidepressant. So based on previous studies, the authors theorized that ketamine works like this. First, ketamine goes through many reactions in the body, and as the body processes it, one product created is known as H&K, or hydroxynorketamine. H&K then interacts with a protein known as EIF4E. EIF4E is a type of protein that helps control what other proteins actually do get made in the cell. Ultimately, the proteins that the cell makes can influence the cell's behavior, and when enough cells change activity, there can be a reduction in suicidal thoughts and depressive tendencies because the circuitry of the brain gets changed. To prove this theory, they had to prove each part independently through experiments. The first experiment involved establishing that ketamine and H&K both result in antidepressant effects. The next set of experiments went to prove that both EIF4E and H&K are needed to cause the antidepressant effect. Simply having one of them isn't enough to cause a reduction in depressive behavior. But when you put them together, that's when you can see the change in behavior. They tested this using an animal model, in this case mice, to analyze behavior. Although mice are different from humans, there are many neurological similarities, so we can test them using an ethical manner. A mouse can't tell you if it's depressed, but we can analyze its behaviors for signs of depression. One method of examining this is called the forced swim test. Here we place a mouse in water, which isn't something it really enjoys, but at the same time isn't directly harmful. And we measure how long the mouse tries to escape the situation. When the mouse is not moving, we call it immobility. The longer a mouse is immobile, the more signs of depression or susceptibility to depression are seen. So for an antidepressant to work, there would need to be a reduce in immobility. Now, in this experiment, we injected some mice with saline, a negative control, some with ketamine, a positive control, and H&K. They ran this experiment and measured the time spent immobile. They found that there was a drop in immobile times for H&K and ketamine, both confirming that ketamine works in mice and that H&K is the reason why it's working. They can now run experiments to see whether H&K really does interact with EIF4E. Now, EIF4E is a substance created by the body. We simply cannot extract it from a mouse. We need to stop its production. So they used a method called transgenic mice, which stopped the gene EIF4E from working in the body. What they found was that the drop we found in the last slide was no longer there when we injected a mouse missing EIF4E with the same things. This now essentially proves that EIF4E is necessary for H&K to work. So to wrap things up, the antidepressant actions, as measured by the forced swim tests, show that ketamine and EIF4E work together to reduce depression. In terms of cell-specific translation, the authors used the research I've explained so far and conducted variants of it to find more information on the mechanism of action. They found that traditional antidepressants have a different route of action than H and K, that EIF4E behaves differently in different cells, and that the differences in proteins resulted in differences in rates of cell communication, creating larger behavioral changes. Thank you all for listening.